All right, thank you. Let's get started. So thank you for joining us today for today's exciting talk by Dr. Patrick Shi on the role of Nav1.9 in human sensory neurons. There is tremendous interest on the potential for Nav1.9 as a new target for pain. Nav1.8 as a target has been validated by the FDA approval of Vertex suzetrigine, successful phase 2 clinical development of Latigo 1, as well as new Nav1.8 inhibitors from Lilly with successful phase 1 trials and others that are now going into phase 1. Nav1.9, on the other hand, has been a tantalizing target who as a drug target has only in the past few years been aggressively explored at levels similar to Nav1.7 and Nav1.8. The next slide, please. First, I want to thank Gary Watkins, Director of Marketing at AnaBios, for putting together this webinar series. As a quick intro for those who have not joined us in the past, AnaBios is a unique contract research organization and biotech company based in San Diego. We recover human organs from ethically consented donors and use the tissues or cells to conduct physiological assays in which we can test preclinical compounds for drug discovery projects. We have essentially redefined first in human, and working with AnaBios is as close as you can get to a clinical trial without actually doing a clinical trial. The goal is to increase the success rate of clinical trials by improving the human translational evidence on moving therapeutics from early stage to late stage and to IND submission. For those developing therapeutics for pain, AnaBios has been evaluating client pain therapeutics for over 15 years. Dorsal root ganglia as well as trigeminal ganglia are recovered from post-mortem donors with both normal condition or pain or neuropathic pathology. AnaBios provides services for the entire drug development pipeline from target identification and validation through the provision of tissue samples for transcriptomics or proteomic work or validation of sensory neuron modulation by tool compounds, to lead generation through supporting medchem or evaluation of gene therapy constructs, and then finally through lead optimization and candidate selection. Finally, a couple of points about the webinar. First of all, there are handout PDFs regarding AnaBios services that can be downloaded from the handout section in the webinar control panel. It should be, if you see up on the top, there's a symbol for going to the handouts. Regarding questions, we'd like to hold addressing questions until the end of the webinar. To submit the questions, please use the chat box in the webinar control panel. You can type in the questions at any point, and I will read the questions at the end of the presentation. So next slide, please. It's my great pleasure to introduce today's speaker. Dr. Patrick Shi is an expert biophysicist and electrophysiologist with over 15 years of dedicated experience in ion channel pharmacology and preclinical drug discovery. He is currently serving as senior scientist at AnaBios, and he leverages high-resolution electrophysiology and human primary tissue models to investigate complex drug-target interactions. He did his postdoctoral training at both Stanford University and Genentech, where he contributed vital mechanistic insights into neuronal, cardiac, and pain targets, including NAV, TRP, HCN, and hERG channels. He's built a distinguished academic portfolio featuring prominent publications in leading journals such as Neuron and JGP. Please note that the work that's being presented today has been submitted to bioRxiv and is in process for submitted to a peer-reviewed journal. Patrick?
All right. Thank you, Richard, for the introduction. And I want to thank everyone for joining today's webinar. In this webinar, I'd like to discuss how we at AnaBios helps clients advance their drug discovery by providing a way to test efficacy and safety in human at a very early stage. And as a case study, I will show how we have developed these capabilities to study a new potential pain target. And here we're particularly excited about these new data about the biophysical characterization of the Nav1.9 sodium channel in native human trigeminal and dorsal ganglia neurons. Now, before I begin, again, like Richard has introduced what AnaBios does, we specialize in conducting translational study utilizing human tissue for transplantation. When an organ becomes available for research, it comes to us with full donor history like age, sex, medical background, and cause of death. And really, the critical step is the second one here. We use proprietary preservation method to maintain functional tissue during the shipping to our laboratory in San Diego. And every sample is QC validated upon arrival before any study begins. And that matters for a practical reason. Functional data such as contractility or electrophysiology and drug response data all requires living and responsive tissues. And if viability is compromised, we don't see the experiment fail visibly. It'll give you an answer that looks okay, but it isn't. And the volume matters too. We have a larger network, which means that we can run multiple donor on the same study, which lets you separate a real drug effect or from a donor to donor variability. And the output is then a functional human pharmacology with documented provenance in a form that you can then include in a regulatory package. So at AnaBios, we recover a range of human tissues, and these are the therapeutical areas that these tissues support. Across the top row here, you can see we procure tissues from peripheral nerve, lung, heart, kidney, colon, and skin, these 6 organ systems, all from human donors. The blue band here is the efficacy work that we work in. For example, we work in pain for peripheral nerve, fibrosis, COPD, and asthma for lung disease, atrial fibrillation, heart failure for cardiovascular, polycystic kidney disease, and renal fibrosis for kidney, and then primarily IBD for gastrointestinal, and itch for the skin. And the gray band here is safety. So we can also assess cardiotoxicity in human cardiac tissue and neurotoxicity using nervous system tissue. So these are the assessment most programs need before the first in human, and we run them in the same tissue types. And the point of showing this full grid is that this approach is universal across all tissues. Whatever indication that you work in, the question remains the same. Does the compound do what you expect in human tissue? And today, I'll focus on the first column, pain in human DRG and TG neurons. So alongside our functional study, we also supply fixed and frozen human tissue for molecular work, such as transcriptomics, proteomic, and immunohistochemistry. Quality here comes from the same source as everything else. For example, how the tissue is being collected. Our samples are recovered and preserved using method designed to maintain viability, and that translates directly into molecular integrity. And this chart shows the RIN score, the RNA integrity score across the 6 tissues, ours in blue, and commercially available tissue in gray. And the red line here at 8.0 is the threshold generally accepted for a reliable RNA-seq work. So our sample sits above that in every tissue. The practical difference is the largest where it matters the most for this audience. If you look at the spinal cord and the DRG, neural tissue is where RNA degrades the fastest and where the gap is the widest. So before any program commits to the clinic, there are 4 questions that needs to be answered to ensure success. The first is, whether or not the target is actually relevant in human. You have expression, distribution, and function that can all be different between species. A target that drives the phenotype in rodent, for example, may be far less important in human tissue or regulated in a completely different way. Second, does the compound work as expected in human? So potency, selectivity established in recombinant systems or in animal tissues don't always necessarily translate. In native human tissues, the target sits in its real environment with its native subunit, native binding partner, and its downstream signaling intact. Third, how does the compound compare with the current standard of care? And this is a commercial question as much as a scientific one. If you run your compound head-to-head against the standard of care in the same human tissue, in the same assay on the same day, then you have a differential argument that survives scrutiny from partners or investors or regulators. And lastly, what's the right dose for the first in human? The human concentration response data give you an exposure target anchor in human biology, rather than the one that you have to extrapolate across species. And what I want to emphasize is that these 4 questions do eventually get answered, but usually sequentially and sometimes late. Some in preclinical, some only after phase 1, but sometimes not until phase 2, when the cost of a wrong answer might be at its highest. Human tissue studies let you answer all 4 of this before you even commit. Same tissue, same platform in months rather than years, and at a much smaller fraction of the cost. So this is the case for building human data into your package early, and it's what we'll walk through for the rest of this presentation. For the AnaBios pain platform relies on 2 native human tissues. On the left here is the human dorsal ganglia. You can see the luxal fast blue myelin stain, and next to it, the CGRP immunostaining with hematoxylin. And on the right here is the human trigeminal ganglia, which is the relevant substrate for studying craniofacial pain, migraine, or trigeminal neuralgia. The histology profiling shows that these tissues retain their native architecture. And from both of these tissues, we can run calcium imaging assay, patch gland electrophysiology, and have the ability to conduct pharmacology studies on a wide variety of channels, as I listed here. For example, 1.7, 1.8, 1.9, KV channels, HCN, A6, etc., TRP channels. And so what I'll show you today is our Nav1.9 data set, and importantly, a comparison of the biophysical property of Nav1.9 between the DRG and the TG neurons. So AnaBios would predict the potential efficacy of a pain drug candidates using 2 different types of assays. On the left here, we're showing the fluorescence imaging-based assay. In these studies, we isolate the human sensory neuron and put them in culture, introduce them to a fluorescence dye, and then the fluorescence responds when a neuron is activated. On the right side, I'm showing an example of data generated using patch gland electrophysiology. These assays are considered the gold standard for studying neuronal pharmacology, and we use them to measure the potency of compounds and also study the mechanism of action. The data from both models are very valuable to our clients and can help them prioritize the compounds with a high probability of success in human clinical trial. And today I will exclusively show data obtained from electrophysiology, but keep in mind that we use both approach when it comes to pain drug discovery. Before we get to the data, I want to spend a little bit of time on why sodium channel matters for pain and why the cell you record from determines what you learn. On the left here is the basic circuit. You have a noxious stimulus at the skin, for example, heat, cold, or mechanical. This is transduced by a peripheral terminal. That signal travels to the cell body in the dorsal ganglia, and from there into the dorsal horn of the spinal cord and then up to the brain. And the DRG neurons is the gateway. If an action potential doesn't fire here, the signal never reaches the brain and there's no pain perception. And that is what makes the DRG such an attractive point of pharmacological intervention. And it is the tissue that we recover from in human donor. On the right here is what governs the action potential actually fires or not. We have three main voltage-gated sodium channels that dominates the nociceptive excitability, and each occupies a distinct part of that waveform. Nav1.7 carries a fast TTX-sensitive current that sits at threshold. It amplifies the subthreshold depolarization and helps bring the membrane to the takeoff voltage. Its role is supported by human genetic, where loss of function mutation produces congenital insensitivity to pain. Nav1.8 carries a slow TTX resistant current, and it is the principal contributor to the upstroke and overshoot of the action potential itself. Nav1.8 is now clinically validated, as Richard has mentioned. Suzetrigine was approved in January 2025 as a non-opioid analgesic. And lastly, Nav1.9 carries this also a persistent TTX resistant current that activate near the resting membrane potential. But rather than shaping the spikes, it's thought that it sets the baseline of the neuron that starts from. So potentiation of Nav1.9 can depolarize the membrane closer to threshold, priming the cell to fire. And that makes it a target of real interest in inflammatory pain, for example, where sensitization rather than acute firing is the driver. And Nav1.9, as the figure indicated, is the least characterized of the three sodium channel and is the subject of what I'll show you today. To further emphasize the importance of studying human neurons, I want to revisit the story of Nav1.7 and Nav1.8. So about 15 years ago, Nav1.7 received a lot of attention and was the target of many drug development programs. In the prior years, several groups have reported that individual with congenital insensitivity to pain carries a mutation that completely inactivated this channel. And therefore, it prompted many pharmaceutical companies to initiate the development of drugs that could accomplish inhibiting or silencing of Nav1.7. But however, there are other companies that decide to pursue a completely different path targeting a completely different channel, Nav1.8, which has also been shown to upregulate during inflammation-related pain. And here's the electrophysiology recording in human DRG of Nav1.7 and 1.8. You can see that with a small current injection in a baseline condition, we can elicit a train of action potentials at a baseline here. Selective blockers such as PF771 against 1.7, and we can test also Nav1.8 using VX548. But you can see that these two compounds shows completely different effects. While inhibition of Nav1.7 shows almost no significant impact on excitability of sensory neuron, the inhibition of Nav1.8 can silence these neurons. And this is highly predictive of Nav1.8 inhibitor working as analgesic in the clinic, especially in the condition of inflammation-related pain. And indeed, FDA has approved this VX548, the Vertex drug, for the treatment of pain. While on the contrary, several Nav1.7 blockers that were tested in clinical trial all failed to show analgesic efficacy. So here are the three goals that we want to address when we started this research project. And the first one is to establish a robust and reliable method to measure Nav1.9 currents in human DRG and TG neuron and generate a culture condition that we can experimental condition that we can do that. Second is once we establish that, then we can compare whether or not there's any tissue specificity and look at the biophysical properties in these two tissues. And lastly, to assess the role in neuronal excitability in normal as well as pathological states. A word on what we actually know about this channel structurally. Nav1.9 has the canonical voltage-gated sodium channel architecture with 4 homologous domains with 6 transmembrane segments in each, with S4 helices serving as the voltage sensor. You have the classic IFM motif in the domain 3 and 4 linker, that is the inactivation gate, and the serine at position 355, that's the key determinants for its TTX resistance. Till this day, however, there is no known experimental structure for Nav1.9. There's no X-ray crystallography, there's no cryo-EM structure. What you see on the left here is just an AlphaFold prediction from the protein sequence. Nav1.9 has resisted structural determination for the same reason it had resisted electrophysiology. It is difficult to express in heterologous system and difficult to record from. And here's the point that matters the most for drug discovery. Nav1.9 is the least conserved sodium channel across species. So human Nav1.9 is only about 75% identical to the rodent ortholog, considerably more divergent than Nav1.7 or 1.8. And that number has a direct consequences. So for a channel this divergent, pharmacology that established in rodent neurons cannot readily be assumed to transfer to human. And so extrapolation can be genuinely risky here, which is why we want to set out to characterize this channel in a human neuron directly. The human genetics for Nav1.9 are also unusual and worth understanding. The channel diagram maps the disease-associated mutations onto 4 domains colored by the phenotype. And what's striking is the range. You have mutation that causes small fiber sensory neuropathy, familial episodic pain, painful peripheral neuropathy. And also in green here, congenital indifference to pain. The last one here is quite paradoxical. You have gain of function mutation in the same channel that can produce either a severe pain or a complete absence of it. The explanation, well, one possible explanation is a matter of degree. So as we all know, Nav1.9 is thought to carry this persistent current that sits near the resting membrane potential. So mutation that enhances this channel can then depolarize the neuron. So it's thought that mutation that causes a modest depolarization brings the cell closer to the threshold and it becomes hyperexcitable, and that phenotype is pain. But a large sustained depolarization can hold the membrane at a potential where other sodium channel begins to inactivate. And when that happens, the neuron can no longer generate an action potential at all, and the phenotype is insensitivity to pain. So same direction of effect on the channel, but opposite outcome for the patient, depending on the magnitude. So for drug discovery perspective, this is encouraging on 2 counts. It confirms that Nav1.9 activity is causally linked to pain in human and not inferred from animal models. And it also demonstrates that the human nervous system tolerates this loss of Nav1.9 signaling, and it's a strong argument for Nav1.9 being a potential analgesic target. So Nav1.9 channel can also be modulated by toxins and inflammatory mediators such as histamine, prostaglandin, and bradykinin, for example. They act at G protein coupled receptors on the sensory neurons. These compounds activate the PKC pathway, which eventually phosphorylates Nav1.9 directly, and the result is an enhanced persistent current. The consequence is that Nav1.9 behaves differently in inflamed tissue compared to healthy tissue. It is not a static channel. It is a channel whose contribution to excitability can increase precisely under condition where pain matters clinically. And the panel on the lower left here makes a related point from a different direction. So Nav1.9 is pharmacologically tractable. There's a range of toxins and small molecule that can modulate it in both directions. Spider and scorpion toxin that can, for example, augment current density and shift activation. but then you can also have compounds that reduces current. And so this channel seems to be druggable, but what's missing is the ability to study properly in human tissue. And so taken together, this target seems to be sensitized, potentially sensitized in a disease state, one that responds pharmacology, and that is a strong case for Nav1.9 as an analgesic target. So I said earlier that Nav1.9 is difficult to record, and this slide shows why and what we did about it. So one of the big problems we face when recording Nav1.9 is problem with space clamp. So it seems that Nav1.9 is expressed heavily and readily in neurites, not just in the soma of the cell. So when we patch the cell body of the neuron with very extensive neurites network, we cannot maintain a proper voltage control over membrane that is distant from our recording pipette. And so the current arises from those distal compartment escape clamp. As you can see from this figure here with the red trace, notice the abrupt all or none current jumps. And so these are the unclamped currents from the neurites. So it's not a graded activation of the channel. And here you can see from the RAM protocol, this abrupt current that's from the distal neurites. Data like this cannot be used for kinetic studies or for voltage dependence or for IC50 determinations. It's just contaminated. And so our solution was to control the culture condition to minimize this neurite network. So on the top row here is TG1, so beta-3 tubulin staining, immunostaining of neurites. And so this is culturing our standard normal culture with GEM21 and growth factors. And you can see by day 5, you see this dense neuron network. And the bottom here, what we did is to culture the neuron without growth factors and culturing at 28 degrees. And you can see less than 24 hours, and even by day 3, there's very minimal neurite network. And by day 5, you can see there's dramatic reduction in the neurite network. And so from here, from these neurons, we can generate this very nice graded activation of the Nav1.9 current. And in the RAM protocol, you can see this very nice clean current. And so this is considered as a well-clamped current. And so current recorded from this, we can then make quantitative pharmacology and biophysical characterization from. So with the clamp control established, we could characterize the channel properly. So on the left here is the voltage protocol and the raw family of traces. We have activation from a hyperpolarized potential going up all the way up to plus 30 with 250 milliseconds. And also inactivation is done with step protocols, conditioning steps. So the top row here is a family of traces from DRG neurons, and the bottom here is from TG neurons. On the right panel overlays the resulting activation and inactivation curves from both tissues. So two things to know here. One is activation. So if you look at the V half of activation, it's not significantly different between DRG and TG. So essentially the channel activates at the same potential in both tissue. But however, if you look at inactivation, here's where there's statistical significant difference. TG inactivation curves significantly shift more to the depolarized potential relative to DRG. So this gray curve here. And that shift matters because it changes this overlap between the activation and inactivation curve, what we call window current. So you can see that shaded right here. This shift increases the window current. And so the window current is where a persistent non-inactivating current can flow at a steady state. So a larger window current means that there's more standing sodium conductor near rest. So we predict from this data that TG neuron, there might be a larger and more sustained Nav1.9 current, and why we expect Nav1.9 to make a little bit more substantial contribution to the resting membrane potential and therefore excitability. So while data support the potential importance of Nav1.9 in both neuron type, it raises the possibility that Nav1.9 modulator could show greater efficacy in, for example, craniofacial pain in TG neurons. So the previous slide was about where the channel operates on the voltage axis, and this one is describing how fast. So we fit exponential to 3 kinetic processes: activation, fasting activation, and deactivation. And so the top row here shows the representative fits. The bottom plots the time constants against the range of voltages. And start with activation on the left. So the arrow marks the voltage range that matters from about -30 to 0 millivolt near threshold. Across that range, you can see that TG neuron shows consistently smaller time constant, which suggests that the channel activates faster in trigeminal neurons than that in DRG neurons. Fasting activation in the middle are essentially superimposable between the two tissues, and likewise, same with deactivation. So the difference here is quite specific. It's not a global shift in channel kinetics. It's localized just to activation and localized to the voltage range where the neuron actually sits when deciding to fire. So combining this with the activation shift from the previous slide, the picture is consistent that in trigeminal neurons, Nav1.9 activates faster and carries a larger window current, and both point to the same direction, that there's a larger contribution to excitability in TG rather than DRG, and there might be a tissue specific difference in Nav1.9. So next, we want to assess the pharmacology of Nav1.9 in human sensory neurons. So this is recorded in human DRG neurons. And the pharmacology assay that we used is described here. We first hyperpolarize to -100 for 1 second from holding potential of -80, and then we depolarize to -20 to activate the channel every 10 seconds. And to validate this assay, we used the recently identified and characterized Nav1.9 selective compound, the icogen 604025, and ran a full concentration response curve. So on the left here, these are the raw traces, the baseline orange, and we did 4 concentrations from 10 nanomolar up to 10 micromolar. And you can see this graded concentration dependent inhibition of this persistent current with essentially a complete block at 10 micromolar. On the right here is the resulting curve. Fitting of the Hill relationship yields IC50 of 560 nanomolar, a Hill slope of -0.7, and R square of 0.9. These data validated the recently published potency for this compound using the recombinant Nav1.9 in stably expressing cell line. But what's important is that for a channel that until recently was considered too difficult to record reliably, that we can actually produce this data format at all in this native human sensory neuron. A well-fitted concentration response curve with a defined Hill slope and IC50. And these are the data that you needed to rank compounds, to compare series, and to support a regulatory package. And so now that we have this capability for Nav1.9 in human neurons. So earlier I showed you that the signaling pathway by which inflammatory mediator converges on Nav1.9 channel, and that is the prediction tested directly in human neurons. So we applied an inflammatory soup consisting of histamine, prostaglandin, serotonin, and bradykinin, and you're recording the same cell before and after. So on the left here is a raw family of traces, and you can tell immediately that the current is substantially larger under inflammatory condition. And the IV relationship in the middle quantifies it. You have 2 changes, and both matter. So first is the peak current increase indicated by the black arrow. It roughly double the peak of the IV. And second is the curve shifts to the left towards a more hyperpolarized potential. So the channel is not just carrying more current, it's carrying it at voltages closer to rest. And the panel on the right is the functional consequence. So this is a RAM protocol, and under inflammatory condition, the current is larger and activates earlier along the ramp. And that is a channel contributing more to excitability under exactly the condition that produce clinical pain. And this mirrors what I've been described for Nav1.8. 2 TTX resistant current, both sensitized by inflammation and both converging on increased nociceptive excitability. So for a drug discovery program, the implication is direct. If you screen at only baseline, you may substantially underestimate what your compound does in a disease state, which is why our pain models include inflammatory sensitization as a standard condition. So the previous slide shows that inflammatory mediator makes Nav1.9 currents bigger. This slide shows something more important. They also make the channel easier to open. And on the left are the curve describing when the channel opens and when it shuts down the voltage dependence, control in black and gray, and the inflammatory in red. So under inflammatory condition, both curves move to the left, but activation shifts considerably more than inactivation. And you can see from the right panel, the cleanest way to see it. These are pair recordings, same neuron measured before and after, adding inflammatory mediator. So now the channel opens at voltages roughly 12 millivolt closer to the neuron's resting state. So in plain term, inflammation makes the channel switch on sooner, the neuron sits closer to firing, and it takes less time to push it over, or less current to push it over. And that's the sensitization that you want to see from a channel implicated in inflammatory pain. And we've shown that here in human DRG. And the practical implication is that a Nav1.9 channel in a healthy tissue and one that in an inflamed tissue are behaving differently. And so if you screen your compound only under healthy conditions, you're testing against a version of the target your patient doesn't have. And you might end up ranking a series incorrectly or pass on a compound that would have worked. And again, that's why we included inflammatory condition as our standard. If you Nav1.9 is your target, the disease state of the channel is the one that may matter. And human sensory neuron are where you can screen against it. So to summarize, Nav1.9 has been shown to be difficult to record. We developed a robust method controlling neural outgrowth for clean voltage control, extending it to measurements in trigeminal ganglia, which to our knowledge has not been characterized before in native human. Second, TG and DRG neuron are not equivalent. There seems to be some tissue specificity. Nav1.9 activates faster in trigeminal neuron, has a larger window current. So if your program targets, for example, craniofacial pain or trigeminal neuralgia, the tissue you choose does matter. Third, inflammation sensitizes the channel. So current increases and the channel opens closer to the resting membrane potential. The channel in your patient is not the channel in healthy tissue. And lastly, we can run quantitative pharmacology, full concentration response curve defined IC50 in native human neurons. And since Nav1.9 is only 70% conserved between human and rodent, animal pharmacology might be a weak guide for this target. So if you have a Nav1.9 program or are considering one, then we can definitely support that. So lastly, I want to touch a little bit on why human data is important here and why Leader has chosen human data. So what I've been describing in my presentation is a translational research done in human tissue rather than in human. So this is not modeling any human biology, but measuring directly in human neuron. Using this with Nav1.7 and 1.8 as an example, these human neurons can actually distinguish a target that worked from the one that did not, years before the trial's readout. With 1.9, human neuron opened a target that most companies had written off as inaccessible. So this strategy is not unique to ion channels and it's not unique to pain. It's definitely a general approach and why the industry is moving in using native human neurons. So for 5 reasons. One is regulatory. So FDA now supports reducing animal testing and using human data to strengthen the IND package. And the second is the cost. So a failed phase 2 clinical trial costs an order of magnitude more than a human tissue study. And for investors and partners, human data carries definitely more weight in diligence. And also, for differentiation, your compound against standard of care in the same human tissue is an argument competitor cannot easily match. And lastly is the review quality. So when the human data is already in the IND package, the regulatory review can move faster and smoother. So Nav1.9 was just today's example. The approach applies to whichever target is on your critical path. And so I want to end it here, and I want to thank everyone for your time, for listening to and joining this webinar. And I'm happy to take any questions. Thank you.
Thank you, Patrick. We have a series of questions that are coming up. To reiterate, if you want to ask questions and you haven't done so yet, there is a button at the top that has a question mark. That's where you type in the questions. Okay, so there are quite a number of questions, as I mentioned. Some of them are somewhat related, so I'm going to take the liberty to somewhat include questions that are related but from different individuals.
Okay.
So first question that I want to ask is there's questions regarding one, how did you isolate Nav1.9 from Nav1.7 and Nav1.8? In that regard, the other question is coming up, which is, how do we know that inflammatory cocktail, the data that you're showing of an increase in the current, is actually not an effect on 1.8? So let's start with, that's a combined question.
Yeah, so here are the recording conditions that we use for these voltage clamp experiments. So we take very careful of isolating 1.9 by using a cocktail of different blockers. So we have nickel, cadmium, cesium, Ta, 4-Ap, block all kinds of calcium channels and potassium channels. We use a saturating amount of TTX and VX548 to block out 1.7 and 1.8. We also supplemented GTP gamma S to enhance the 1.9 current, so we can reliably see the 1.9 current in all our cells. So we're quite confident that what we're measuring is 1.9 current, and so we've using these recording conditions, the current that we see are the isolated Nav1.9, and again using the selective inhibitor, the ICA 604025, we validated that obviously we validated the compound works, but also we validated that what we're measuring is Nav1.9. So therefore, the inflammatory soup condition that we see with the increase in current, it would be affecting Nav1.9 current. Now, does the inflammatory affects 1.8? Probably does, but in our data here, because we have now cleanly isolated Nav1.9 currents, the effect that we see is Nav1.9.
Okay, thank you. Before we're proceeding to all the questions about electrophysiology and physiology, one other question regarding more towards the biophysics is the reported difference in V-half of activation from previously reported recordings of NAV 1.9. So it's specifically roughly an estimated 20 millivolts. So can you comment on that?
Yeah, so one of the things that I think a lot of the literature uses fluoride as the internal anion, and fluoride can activate G protein pathway and causes a left shift in the voltage dependence. So this is the exact reason why we use cesium methensulfonate as the primary anion, and we only have a small amount of cesium fluoride just to maintain a stable membrane seal. So so that's why recording in Nav1.9 using the correct internal anion is appropriate. Using methensulfonate and chloride might be a more physiological approach, whereas fluoride will give you a much more left shifted, about 20. Yeah, it's exactly about a 20 something millivolt left shift in your voltage dependent inactivation, for example. But what you get in that benefit is that you can hold the current, you hold the cell at minus 140, for example. But it's definitely not physiological.
Okay, thank you. Just one other kind of biophysical question. It would appear that, at least from the tracing, that inactivation was faster in the DRG versus the TG.
Yeah.
Is that, yeah, I think you mentioned this, but can maybe reiterate that?
Yeah, so we noticed that there are variability in the current traces. So it looks like in that sample trace, the inactivation is slightly faster in the TG and DRG, but because there's so much variability and error, that it might not be representative. So there's some TG cells that has very slow, some that are fast. So there's quite a lot of range in that. So so to be honest, I think there aren't that much difference between the two tissues in the fast inactivation.
Okay, thank you. Regarding the window currents specifically in trigeminal, so this potential increase in window currents, are they actually sufficient to cause or mediate inactivation, somewhat similar to observed with just gain of function mutations? In size, shall we say?
You mean like if there's any mutation that can cause this?
No, I meant more like, is this increase in window current that is there I mean, there's a difference in the conductance clearly. Does that cause an increase in window currents in TG that could mediate inactivation? Somewhat similar to, you say, the question is with regard to mutations that have been observed.
Yes, I would think so. If, for example, the window current is large enough, then for example, a mutation that can cause a large shift in the inactivation, and then that large window current can probably cause a depolarization and inactivation of other channels as well, if that's the question.
Yeah. I think that addresses the question. So how about another question? So what is the fraction of DRG neurons that seem to have Nav1.9 currents?
Yeah, that's a good question. That's a good question. So I've done these recordings both in GTV gamma S and without GTV gamma S. So with all of these experiments are done with GTV gamma S, and it seems that almost all our cells have certain degrees of Nav1.9 channels. So the current size does vary, which that's not surprising, given that Nav1.8 also varies between cells. But in GTV gamma S, we see a range from sometimes 2 or 3 nanoamps of current all the way up to like 10, 15. However, when I record without GTV gamma S, we do see that Nav1.9 is likely there in all cells, but definitely the expression level is different between perhaps different cell types. So there are some cells that have maybe only hundreds of picoamps of current, and then there are some cell types that has 2 or 3 nanoamps. So it does vary, but and for cells that are maybe a couple hundred picoamps, it's hard to tell if that is 1.9 or leak. And so in those cells, are there any Nav1.9, or does it make a significant contribution? Then I would say that in those cases, likely not. So, but I would say that there is a certain degrees of Nav1.9 in almost all cell type, but it does vary in the amount.
Mhm. So you talked about the space clamp issues, and we have, and in discussions with folks, have discussed the potential of an actual decrease in Nav 1.9 over time in culture. Is that actually still what we think is going on? And what would we
Yeah,
do to mitigate it, like similar to this neurite
yeah. So
stunting?
yeah, that's an interesting point. We haven't really seen a substantial decrease in current over time in culture necessarily. So we've measured in our node growth factor condition at least, um, at less than 24 degrees, and then also at 3 days in culture, and we don't really notice a substantial decrease in NAM 1.9 currents. And so, but are the decrease in NAV 1.9 currents, is it just because the NAV 1.9 is expressing along the neurites? I don't know if it's moving along the neurites. So then now what happens is, if you measure at the soma or the cell body, you have
Hmm.
this massive distal current that can then mask your somatic current. And so
Hmm.
in essence, it appears to be smaller, but because there's so much distal NAV 1.9 that's coming into that you're measuring. So that might contaminate the results a little bit. So it made it seems like maybe the somatic current is less. So we do notice that over days, if it's got a dense network of neurites, the distal Nav 1.9 current is much larger than the somatic. We do see these really large
Hmm.
abrupt currents. So
Hmm.
yeah,
Regarding the pharmacology reporting for the icogen compound, so just as a baseline, what can you comment on the stability of this of the recordings in the absence of compound?
yeah. So here's an example of some stability that we ran with this protocol. So these are just time match controls
Hmm.
of just running this current over a period of time, and you can see that the current is actually quite stable. These
Hmm.
are three sample cells, 33, 34, 14 minutes. So, if there are clients that wants to test compounds, I do suggest that we can test, for example, two concentrations per neuron, and then create a two cohorts
Yeah.
to generate like four concentration curve. But it is quite stable.
Several people have asked this question. What about this compound and current clamp or exponential firing?
Right. So that's definitely in the works right now. So yeah, we're working on that. Um, and so yeah, can't really give an answer right now. But yeah, we're that's what we're working on.
Right. Um, Another note, which is that so the reported pharmacology, shall we say IC50s for resting is, well, ours appear to be closer to the resting IC50 that was reported by GSK and Omniab or Icogen versus the inactivated state. Any comments on that?
Yeah. So yes, R is a resting state protocol. So we're
Mm-hmm.
in the process of developing an inactivated protocol. And so 1.9 does it's proving to be a little challenging designing a stable inactivation based protocol. So we're in
Mm-hmm.
the development of that right now. Yep.
Um, Have you tested this same pharmacology, but now in an inflamed, in the treated with inflammation?
No, not yet. And that's a good point. And so, yeah, that's something that we can look into as well. Yep.
Mm-hmm.
Yep.
So have we done this between normal and a donor that has some reported pain?
No, we did not. So these donors they're not, for example, in chronic pain donors or anything like that. It will be interesting to see what these neurons will look like in donor that has chronic pain. Yeah, no, but we haven't done that.
Back to one biophysical question that I did not ask earlier. I've missed it. What about temperature and Nav1.9?
So all of this is done in room temperature, and yes, so in Nav1.9, the Q10 seems to be much steeper. So it would suggest that in, for example, physiological temperature, like 37 degrees, all of these kinetics will be much faster. And so that kind of brings the question that does the difference between DRG and TG, that difference becomes even greater in 37 degrees. But yeah, that's something that we have to keep that as in mind as a caveat, is that the Q10 for Nav1.9 is a bit steeper, and so if we want to make physiological implications, we probably have to do some measurements at 37 degrees.
Yes. Here's an interesting physiological question, which we haven't really dissected this, but the question is do we think, or can we test a difference between how Nav1.9 contributes to excitability in neurons involved in visceral afferent signaling versus somatic?
We can, we can definitely, I think we can procure tissues from, yeah, that's
it's a challenging question,
yeah.
but the answer is no, because we're currently pooling DRG from thoracic to lumbar. So this would require dissection of, and also potentially getting some other ganglia, but dissecting the specific ganglia and then isolating it that way.
Yeah, I don't think we can separate them out yet right now, but can definitely have to do that.
So can you comment on the cell size for which you are recording these?
Yeah, so the cell sizes ranges from, so we don't pick particularly small cell sizes so it ranges from anywhere between 30 microns up to 60, 70 microns. And so, so we don't see much, for example, variation in, for example, like, okay, only smaller neuron has more Nav1.9 or larger has less. So yeah, so we don't we don't bias any cell size.
So here's another question, again, about maybe more about thought about approach. Have we done any dual block yet of in calcium imaging?
No, not that I know
of. No, we have not.
Yeah.
Here's a question about the protocol. Do you actually have to hyperpolarize to see the current?
No, we don't. But hyperpolarization would appear to increase the current a little bit. So what we want to do is try to push the channel to the most stable kind of resting state without compromising seal. And so holding at -80, you will see some current, but there's definitely going to be a degree of proportion of channel that are still inactivated. So going to -100 will then recover those channel, and generally give you just recover more channel from the stable from the inactivated state, so you get a little bit more current, and it seems to be a lot more stable.
Another question now, focusing a little bit on TG. So there were a couple about this. One was about whether or not we've tested the icogen compound on TG. But you can combine that answer with maybe comments on the prep of the TG compared to the DRG. Essentially, what's the health? What kinds of yields roughly?
Okay, so the first question, no, we have not tested this icogen compound in TG. So, but it's, we're gonna do that once we have the TG neuron available. The second is the prep itself for DRG and TG are identical, and so we don't have a separate protocol just for TG. But in terms of, for example, morphology or density and things like that, honestly, morphologically, there are not so much difference between TG and DRG in culture. Yeah.
Okay. Thank you. I believe. I mean, there have been many questions, and I'm trying to make sure that I did not miss anybody on this. Here's one other question, which is that it has been reported that inflammation has actually lead to a decrease in currents. Any comments about the differences between that work and the work you're reporting?
Yeah. So, I think in that paper, I mean, they just say that take their data with a little bit of caution because of the low N number. And so, our cocktail, one of the things is that I think our cocktail is a little different than theirs. We have a little bit more histamine in our inflammatory soup than maybe they used. So I am aware of that paper. And so I will say that maybe there's some difference in the culturing or the inflammatory soup agent that we use, but the inflammatory effect that we see here is consistent with what seen in rodent and in vivo study, where there's a heightened excitability and pain. So yeah, I was aware of that.
Okay. Thank you very much. We're going to leave it one last question, and I appreciate everybody as we've gone actually over time. So thank you, Patrick, for joining us and answering the questions, as well as people adding, showing the interest and asking a tremendous number of questions. So the last question, somewhat related to the window current, but what's the availability of these currents around typical resting versus the holding at minus 180, not 180, sorry, minus 80. And then and then take into account the inflammation.
Can you repeat the first part? Channel.
So what's the availability of Nav1.9 currents around the typical resting membrane potential, which I believe is more close to minus 60 versus minus 80. And especially when you now think about that with regard to applying inflammation.
Yeah, so we haven't really measured, for example, comparing the current level at minus 60 versus, for example, minus 80. And one of the big problem was to get the stable current, for example, measurement at minus 60, which would represent like a resting state. My prediction is that at -60, you're probably seeing substantial amount of inactivated Nav1.9 channels. So the persistent current at -60, conducted by 1.9 and the resting memory potential is likely very low. And so, but in an inflamed state, my prediction will be that that persistent current is gonna be much bigger at resting memory potential, and so that can become a driver for heightened excitability in an inflamed case. But no, we haven't quantified, for example, how much current is at -60, and then how much current was at -80 or -100 to see how much, for example, inactivated channels are there.
Mm-hmm.
And then the main problem is trying to get a stable current holding at -60, and that doesn't run down and etc. So it's more of a protocol experimental challenge.
Thank you. Actually, one extra question. I lied. How long do these TG preps last in culture?
TG culture, depending on what experiments it's done. Typically, when we're measuring Nav1.9, we want to keep the culture as early as possible to make sure that the neurites doesn't start growing and
Right.
start contaminating our voltage current. However, current clamp you can maybe culture the cell a bit longer and still study them reliably. But voltage clamp, yeah, I will keep it at probably BIV 0 to 3
Yep.
for any reliable voltage clamp recordings. Current clamp, going to DIV 5 and 6 would be fine. But normally we do other channels. For example, not 1.9. I mean, we can culture TGs for a couple of weeks.
Yeah. Thank you, Patrick. And again, I want to thank the audience for your attention and your your interest in the Nav1.9 story that we're doing at AnaBios, and I appreciate it. Again, if you want to read the paper, it's been submitted to bioRxiv and will be submitted again to journal. Again, thank you very much, and hopefully you can join us next time we do another webinar, in particular a pain webinar. Thank you.
Yeah. Thank you, everyone.